Hello, and welcome to the Nucleic Acid Delivery for Disease Therapy presentation by Marise.Habib, Jessica Ryle, and Timothy Trousdale. In this presentation, we will cover the purpose of the nucleic acid delivery for disease therapy, the history and development of the technique, how it works, the applications, advantages, and shortcomings of the technique, and the potential future applications or extensions of gene therapy. The biotechnology of gene therapy has been an interesting field for not only research, but also pharmaceutical and diagnostic products for many years, aiming to prevent, ameliorate, and treat certain diseases. Since James Watson and Francis Crick discovered DNA in the 1950s, we have been since developing a better understanding about how the human body functions and communicates, leading us to the discovery of nucleic acid delivery for disease therapy, to prolong the life of most patients who suffer from hereditary conditions, infectious diseases, and some cancers. Before we delve deeper into how gene therapy works or how efficient it is, let us explore the biomolecule that is the building block of this technique. Nucleic acid is a biomolecule that stores and transmits the genetic information of a cell. It is a chain of nucleotides which are composed of three parts: a base, a sugar group called ribose, and a phosphate group attached to the fifth carbon of the ribose. To keep the nucleotide together, three bonds exist in the chain of these connected units. A phosphodiester bond that connects the third carbon of one ribose group to the fifth carbon of the next ribose group. A glycosidic bond that links the sugar group to the base. And hydrogen bonds that link the bases between complementary DNA strands. Nucleic acids include RNA and DNA. RNA, or ribonucleic acid, is a single-strand nucleic acid with the sugar ribose as a backbone and four bases: adenine, guanine, cytosine, and uracil. DNA, or deoxyribonucleic acid, is a double helix 3D structure that forms when two strands link together through hydrogen bonds between complementary base pairs. Deoxyribose is the backbone sugar, and the four bases are adenine, guanine, cytosine, and thymine. DNA is the genetic repository of the cell, while RNA carries and transmits this genetic information to encode the amino acid sequence specified by at least one gene. Gene therapy is a therapeutic delivery of nucleic acid used to treat or prevent disease caused by a defective gene by inserting the healthy gene into the patient's cells instead of using drugs or surgery. Several approaches to gene therapy have been tested, and that includes replacing the mutated gene causing the disease with a healthy one, inactivating a mutated gene that is not functioning properly, or introducing a new healthy gene to combat or eradicate the existing disease. Gene therapy is used to treat infectious diseases, diabetes, hereditary diseases, many cancers, and inborn errors in metabolism caused by one single base change in the DNA. Gene therapy is used to manage and treat few conditions, such as cystic fibrosis, by delivering the gene to replace the dysfunctional cystic fibrosis transmembrane conductance regulator, or ameliorate the function of the current one that is not functioning properly because of the defective gene. It can also treat hemophilia B, somatic cell, anemia using the gene for erythropoietin, muscular dystrophy using the gene for insulin-like growth factor 1, peripheral vascular disease, the degenerative and fatal disease of the cardiovascular and central nervous system. It is also used for lipoprotein lipase deficiency, for AIDS using the retroviral mediated gene transfer to deliver HIV-1 antisense, and it is also used to treat some cancers. This infographic describes briefly how gene therapy works. The cells are removed from the patient, and then in the lab, a virus is altered so it cannot reproduce. The gene is then inserted into the virus, and then the altered virus is mixed with cells from the patient to become genetically altered, and then these cells are injected back into the patient to produce the desired protein or hormone. Safety and specificity are crucial in gene therapy, as there are a few potential risks, disadvantages, and misuses. The use of insulin-like growth factor 1 gene that repairs and bulks up muscles, and the use of erythropoietin gene that increases the oxygen carrying capacity in the blood are two misuses of gene therapy mostly known among athletes.
History and development. The concept of nucleic acid disease therapy has been around for decades. In 1944, the idea of transferring genes within nucleic acids was theorized by Avery, McLeod and McCarty. 2 decades later, Joshua Lederberg worked towards the concept of correcting defective cells by introducing new genes. Stanfield Rogers was the first to transfer foreign genes purposefully into a human by injecting a virus found in rabbits, which normally causes a hard growth to help combat an enzyme deficiency presented in siblings, which was ultimately unsuccessful. Martin Klein attempted to treat a hereditary blood disorder. Klein took bone marrow cells and transformed with plasmids carrying specific genes and a herpes virus to improve the defective marrow cells and returned it to the patient. While these 2 trials were ultimately unsuccessful, they have allowed the development of guidelines allowing for further ethical research. In 1989, in order to treat patients with advanced stages of melanoma, tumour tissue was removed and tumour-infiltrating lymphocytes were introduced, grown and underwent retroviral transductions. The resulting retroviral genes were intravenously administered to patients. This allowed for 2 out of 5 patients' cancer to regress. This was the first approved study to allow genetic material to be administered. A breakthrough that occurred during the 90s was the eradication of rabies in France and Belgium. This was achieved by administering oral vaccines to foxes, raccoons and coyotes. While this was animal-based, the technology of this vaccine led to the advancement of various human-based cancer and disease vaccines found today.
The advantages of gene therapy have become more prominent over the years with the advanced research in biotechnology. Gene therapy can reduce the rate of surgical procedures required to treat or manage certain conditions, such as cardiovascular diseases, when targeting the right host cells. Gene therapy is less intrusive, and therefore the risks and adverse side effects of this therapeutic technique can be avoided. It is a relatively safe technique, but it requires high specificity and accuracy to achieve safe and efficient outcomes. Last but not least, gene therapy gives hope to patients who didn't have any before and improves their quality of life, especially for patients who suffer chronically.
Disadvantages of gene therapy include social cleansing, religious considerations, ethical considerations, problems with cell divisions, recovering from the virus naturally. Gene therapy causes more problems. Whilst gene therapy has emerged as a promising strategy for a wide range of inherited and acquired diseases, due to the poor cell uptake and biological instability of nucleic acids, their delivery has been performed using either viral or non-viral vectors. Although there are different viral and non-viral vectors for gene therapy, all gene therapy applications depend on the fact that genetic material needs to be delivered across the cell membrane and ultimately to the cell nucleus. There are two types of gene therapy aiming to achieve this: germline gene therapy, which is currently not in use, and somatic gene therapy. There are three types of somatic gene delivery. Ex vivo delivery occurs when genetic material is explanted from the target tissue or bone marrow, cultivated and manipulated in vitro, and then transducted and/or transfected into the target tissue. In situ delivery is the administration of the genetic material directly into the target tissue. In vivo delivery is the transfer of genetic material through an appropriate vector, which can be viral or non-viral, into the target tissue. Viral vectors used for gene delivery include, but are not limited to, retrovirus, which can transfect dividing cells because they can pass through the nuclear pores of the meiotic cells. Adenovirus types 2 and 5 can be utilized for transferring both dividing and non-dividing cells and have low host specificity, so they can be used for gene delivery into a large range of tissues. Adeno-associated virus are like the adenoviral vectors in their features, but because of having some deficiency in their replication and pathogenicity, are safer than adenoviral vectors. Herpes simplex virus delivers transgenic DNA, and because of its neuronotropic features, it has the greatest potential for gene delivery to nervous systems, tumours, and cancers. Poxvirus are widely used for high-level cytoplasmatic expression of transgenes. The insertion of the transgene sequences is somewhat different from other vector systems and utilizes homologous recombination or in vitro ligation for construction of recombinant vaccinia virus vectors. Lentivirus are a subclass of retroviruses. They've recently been used as gene delivery vectors due to their ability to naturally integrate with non-dividing cells, which is a unique feature of lentiviruses as compared with other retroviruses, which can infect only the dividing cells. The slide depicts a simplistic diagram of gene delivery viral via vector from the initial stages of DNA being incorporated into the virus to the final step of protein synthesis. Non-viral vectors used for gene delivery include, but are not limited to, naked DNA, which is also known as plasmid DNA or pDNA, and that alone is able to transfer a gene. Its efficiency for gene delivery is low, so it is used for some applications such as DNA vaccination. DNA particle bombardment by gene gun is when particles are coated with plasmid DNA and then accelerated to a high speed by pressurized gas to penetrate into target tissue cells. Electroporation is temporary destabilization of the cell membrane targeted tissue by insertion of a pair of electrodes into it so that DNA molecules are able to penetrate into the cytoplasm and nucleoplasm of the cell. Hydrodynamic is a simple and highly effective method for direct intracellular delivery of any water-soluble compounds and particles into internal organs. Ultrasound can make some nanomeric pores in membrane to facilitate intracellular delivery of DNA particles into cells of internal organs or tumours. Magnetofiction has the advantages of the non-viral biochemical and physical transfection systems in one system. It uses magnetic fields to concentrate particles containing nucleic acid into the target cells. Magnetofiction has been adapted to all types of nucleic acid. Chemical non-viral delivery systems are nanomeric complexes, which include compaction of negatively charged nucleic acid by polycationic nanomeric particles belonging to cationic liposomal or cationic polymers. Cationic liposomes have unique characteristics such as capability to incorporate hydrophilic and hydrophobic drugs, low toxicity, and targeted delivery of bioactive compounds to the site of action. Cationic polymers, as the molecular weight of the polymer increases, the net positive charge of it also increases and therefore are able to bind DNA tighter and form more stable complexes. Lipid polymer systems are three-part systems in which DNA is first pre-condensed with polycations and then coated with either cationic liposomes, anionic liposomes, or amphiphilic polymers with or without the help of lipids. A simplistic diagram of the steps for non-viral gene delivery from initial creation of the lipoplex to the final stage of protein synthesis. And this diagram also demonstrates the gene delivery by physical methods as seen in the bottom right. Ethical dilemmas overshadow germline therapy, with questions such as what diseases or disabilities warrant the use of gene therapy, and when does a trait become a disability? These questions remain both relevant and unanswered. Somatic gene therapy, which is currently being researched and applied, focuses more on techniques that are used and the shortcomings within those techniques. In ex vivo delivery, the technique is used in cases where the target cells act as protein secretion resources, causing major limitations, and at present only a small percentage of reimplanted cells remain viable. In situ delivery, low efficiency of transduction is the main problem because in cancer therapy, one malignant cell can re-establish the tumour again. In vivo delivery, the problem is insufficient targeting of vectors to the correct tissue sites.
Current applications. There are a number of applications for gene therapy. Different delivery methods are appropriate for different diseases. Cancer. The vectors most suited in the treatment of various cancers are viral. As viruses are notorious for infiltrating host cells, they are well equipped to deliver gene-altering nucleic acids. The downside to viral vectors is the immune response to them, diminishing the ability for multiple administrations. As the goal is regression for cancer, this is not an important factor. Parkinson's disease, another viral vector that allows genes to be implemented in brain cells to produce enzymes required to synthesize dopamine. Traditional drugs have little effect on this disease due to the inability to cross the blood-brain barrier, which is why gene therapy is a practical option. Synthetic vectors are man-made delivery systems that attempt to match the efficiency of viral vectors without inflicting an immune response. This would allow repetitive treatment. A current downside to synthetic vectors is the poor packing and unpacking of nucleic acids once the targeted cells are reached. In the case of cystic fibrosis, synthetic vectors have proven to be advantageous over viral options. Lungs have a heightened immune response due to the nature of regular exposure to external invaders. Cystic fibrosis is a prime example of how a defective gene could be altered by introducing a functioning gene to cure or prevent a disease. 9 patients with haemophilia B were treated using a non-viral vector. The delivery of DNA promoted the production of factor IX, a protein used to form clots. One dose was sufficient for individuals to cease their usual treatment of clotting infusions for 2 months to a year. 2 of the patients recorded mild immune response to the treatment. However, that was manageable with steroids. This is a significant decrease compared to previous gene therapies.
All gene delivery systems have shortcomings, and many of these shortcomings narrow the use of different individual methods on the types of tissue and diseases they can treat. When generalizing, the viral vectors have only short-term effects, questionable stability, and the production of these vectors can be quite difficult. Generalization of the non-viral vectors, which include both physical delivery and chemical delivery, is that they have low efficiency acute immune responses, especially present in chemical delivery, and a risk of tissue and/or organ damage.
Future applications. Gene therapy for the renal system has not seen the same amount of attention as other organs. However, a mixture of viral and non-viral treatments could be the answer to simultaneously treating the vast cell types that allow the kidney to function. Therapy for cardiovascular disease is currently being investigated. Cardiomyocytes responsible for heart contractions can slowly diminish in numbers over the years from common issues like hypertension. A myocardial infarction, on the other hand, can cause a mass destruction of cardiomyocytes in a single episode, which is irreparable. While trials have been unable to progress to the clinical stage, animal-based trials have seen promising results in regenerating the contractile cardiac muscle. Using stem cells to treat neurodegenerative disorders such as MS have had a positive impact in the field. Beta secreting human bone marrow stem cells have the ability to suppress immune responses, making them invaluable in the area of gene therapy. As the research and continued advancement of efficiency, the use of non-viral vectors will continue to grow, with superior cost-effectiveness, reduced immune response, and limitless sizing. Non-viral effectors appear to be the way of the future.